Hey guys, Dr. B. This is just a video to help you with X chromosome inactivation in more detail. Just what you've been looking for, I'm sure. So basically, here's an embryo, a female embryo, and as you can tell, it has two copies of an X chromosome, the purple and the blue. And that's what we would expect from almost any embryo. And again, the problem is that, here we have the need to inactivate one of them. And you can see here in this case, it's totally random which one gets inactivated. And again, the reason why is dosage of the gene. We either don't want them to compete. For example, like if this one happens to have, some gene that turns something off and this one has the same gene that turns something on, we don't want them to compete, or we don't want to produce more than the amount of a product that we need. So nature has decided that we need to shut off one of the female X chromosomes so the dosage is right, because remember, males only have one. So this video is kind of like, how does that happen in a little bit more detail? And it's fairly I wouldn't say too complicated, but let's get to it. So this is called the X inactivation center, and on every X chromosome there is this area, and there's these two particular genes of interest that we're going to be dealing with for a second. And the first one is XIST, actually pronounced ZIST, which is kind of funny. And the other one is basically, if you take a look, it's actually ZIST backwards. It's TSIX. This particular gene is going, if this gene is activated, then the X chromosome is going to become a bar body, which means inactive. If this gene is activated, it's going to be the active X chromosome that actually is going to be used to produce proteins throughout the life of this particular female. And so, we talked a little bit in the last video about calico cats and why that happens. So the other thing of interest is that the XIST is transcribed this way and the TSIX is transcribed backwards, and that's a great way to ensure that only one or the other is going to be transcribed. So if we kind of zoom out here for a second and take a look at both of these chromosomes, we see here that we have these pluripotency factors, and pluripotency is just a word that means lots of little pieces of things. And these are all factors that are going to have a role in deciding which one of these things is active and which one's not. So in this case, this is going to be XA, which is going to be X active, and both of them right now look like they're going to be active. But again, one of them has to be shut off. So we need secondary factors that are going to help us to bring these chromosomes together and then jump onto one, and then that's going to be the one that's going to stay active. So CTCF, you don't necessarily know what these things are, but these happen to be transcription factor controls. And the first thing they're going to do is bring together the chromosomes like that. Whoops, sorry about that. There we go. It's going to bring them together. Whoa, I'm drunk driving. There we go. So it's going to bring them together like that. And so now we see that this would be a normal pair of homologous chromosomes in this case. And what they're going to do is, at this point, is a critical point, because right now they're both active. We need to decide which one of these is going to be inactive. And so what's going to happen is all these factors are going to jump onto one or the other and activate whichever one they jump onto. So in this case, you're going to see that the pluripotent factors and CTCFs are going to jump onto the top one. These are going to allow the transcription of TSIX, which again was the green light. So this one's going to be allowed to continue to be the active X chromosome. This one now, because there are no factors for it, are going to have the opposite direction of transcription. ZIST is going to be transcribed and translated through a transcription factor, and that is going to start. And remember, this is the gene that its protein begins the process of inactivating the X chromosome. And so this is the beginning of the production of the bar body. And so all of a sudden now we're going to have all kinds of other factors coming over. Of course, factors you're noticing are everywhere, mixing with these RNAs. And from this point, we're going to get super production of all kinds of ribonuclear proteins. And of course, that's going to draw over more troops. I'm just being general. You don't need to know the extreme detail of it, but the cascade that is going to happen over the next probably about 35 seconds here in this case are going to start to create an X chromosome that's no longer active. And so now we see the running, I like to call it the running of the bulls. These RNAs are going to now attach to the length of the X chromosome. And of course there's other things on there, but I'm just making it simple as it can be, I guess. And then from this point you're going to see that we also have one final step, which is the DNA methyltransferases, in which we know what those things do, if you recall. And most of those actions are silencing of most genes by the enzyme DNA methyltransferase is transferring a methyl group to DNA, which generally is a way to shut it off. So this is a great way for the cell to shut off the length of the entire series of genes on the X chromosome. So that's a great system to control the silencing of a gene. In this case, this chromosome will then shrink and will remain a bar body throughout the life of the organism. There you go.